everyone, and welcome to the final topic for CIE. Topic 19, genetic technology, one of the hardest topics. And in this video, I'm going to go through everything you need to know, much like my A-level notes do as well, which go through all of the theory for the entire A-level, key terms, key marking point, and key examiner points as well. So check that out in the link below if you want that boost to your A-level grade. But for now, let's get into the content. Topic 19, the final topic, is all about gene technology. So genetic technology, and we're starting with the principles of genetic technologies. Recombinant DNA technologies is the technology of combining different organisms' DNA together to create what is known as recombinant DNA. And this is created by artificially joining together DNA of two different species. This is needed for scientists to manipulate and alter genes to improve industrial processes, for medical treatments as well. And then transferring DNA fragments between or within species is possible because the genetic code is universal. Transcription and translation also work the same in all organisms. So what we mean by that is the same triplet of bases code for the same amino acid in all organisms. So you can take the gene from one species, insert it into the DNA of another one, and that same protein will still be created because the genetic code is universal, and protein synthesis, transcription and translation, will work the same way. Genetic engineering is the manipulation of an organism's genetic material, introducing a new gene, and therefore causing that organism to code for a protein that results in the desired traits or characteristics. The process involves precise techniques to modify specific genes within the organism's genome, and one common method in genetic engineering involves the transfer of a gene from one organism to another, resulting in the expression of that gene in the recipient organism. This transfer of genetic material can lead to the production of new proteins or the modification of existing cellular functions, but ultimately it influences the phenotype of the organism. So this is just a summary here of some recombinant DNA technologies: creating DNA fragments, genetic fingerprinting, and genetic counselling. And we're going to be looking at this branch first of all, how you can create DNA fragments to do genetic engineering, because all of this bit here in the blue is genetic engineering. So DNA fragments, so the first step in this recombinant DNA technology is to isolate the fragment of DNA that you want to insert into the DNA of another organism. And there are three methods that can be used to isolate these fragments of DNA: reverse transcription, restriction endonucleases, and the gene machine. Reverse transcription, this is when an enzyme makes DNA copies from mRNA. And this enzyme naturally occurs in viruses such as HIV, and it is reverse transcriptase. So what you'd need to do is locate a cell that naturally produces the protein of interest. That would be because that cell will have lots of mRNA for that protein. You can then remove that mRNA and combine it with the enzyme reverse transcriptase, which is the enzyme which is going to join the DNA nucleotide bases to the mRNA sequence. So we can see here we've got the mRNA molecule. You'd have complementary DNA nucleotides aligning opposite those mRNA bases, and then you'd have a single strand of DNA created. Now you'd need to have a double strand. So to make this DNA fragment double-stranded, the enzyme DNA polymerase is then used. And you'd actually then go on to use this in PCR to get multiple copies. But the key things that you need to know is that you start with an mRNA sequence. You then add in DNA nucleotides and the enzyme reverse transcriptase, and that will then create a complementary single strand of DNA to that mRNA sequence. Then you add DNA polymerase to create a double strand. Now, the advantage of this way to create your DNA fragment is the complementary DNA that is made is intron-free because it was created from mRNA, and mRNA has already had the introns spliced out. Restriction endonucleases is another method, and these are enzymes that cut up DNA. And these naturally occur within bacteria as a defense mechanism. There are many different restriction enzymes that have an active site complementary in shape to a range of different DNA-based sequences, because that is what the active site is. It's the parts that is going to be complementary in shape to the substrate, which is a particular sequence of DNA bases. And that section of DNA bases is described as the recognition sequence. And in this way, restriction enzymes or endonucleases cut DNA at specific locations. Some enzymes cut at the same location in the double strand and create a blunt end, which isn't particularly useful because if you have a blunt end, it's really hard to join the DNA back together. But other restriction endonucleases cut to create staggered ends and exposed bases. And that's what we can see here with this restriction endonuclease, HIN3. It cuts and creates this staggered end. And therefore we have what's known as sticky ends, called sticky because you've got these exposed bases which would then be able to align opposite their complementary bases, and in that way it's a bit like they're sticky because they can join together more easily. So you get these cuts at palindromic sequences, meaning it's the same one way as it is backwards, the DNA base sequence, and that's how we get our sticky ends, which can then help to insert that DNA fragment into the DNA of another organism. The gene machine, this is when DNA fragments are created in a lab using a computerized machine. Scientists first have to examine the protein of interest that they want this new organism to produce, and in that way they identify the amino acid sequence of the protein, and from that they then work backwards and work out the mRNA and DNA sequence that would be required to code for that amino acid sequence. That DNA sequence is then entered into a computer and checked for biosafety and biosecurity, that the DNA being created is safe and ethical to produce. The computer then creates small sections of overlapping single strands of nucleotides that make up the gene, called oligonucleotides. Oligonucleotides can then be joined to create the DNA for an entire gene. Once you've done that, PCR is then used to amplify the quantity and to make it double-stranded. So this process using a gene machine is very quick, it's accurate, and you can also design it so it's intron-free. So that's how we create our DNA fragments. Next then we're going to have a look at how you can clone those fragments so you get lots of copies of it. So you could do this in vivo, which means inside of a living thing. So we've talked about how you create your DNA fragment of interest. Now we can look at how you insert that DNA fragment into a vector, which is an organism that will carry the DNA into the host, and the host is the organism whose DNA you are modifying. So once we've actually used those restriction endonucleases to create our DNA fragment, and we've got our sticky ends, we actually have to modify the DNA so it can be used in transcription. So we have to add a promoter region, which is added at the start of the DNA fragment. And this is a sequence of DNA which is the binding site for RNA polymerase, which will therefore enable transcription to occur. And we have to also add a terminator region, which is added at the end of the gene. And this causes RNA polymerase to detach and stop transcription. So only one gene at a time is copied into mRNA. Once we've done that, then we insert this DNA into a vector, and a vector is something that carries this DNA fragment into the host cell. Plasmids are the most common vectors which are used, and plasmids are loops of DNA that are found in some bacteria. So that circular DNA, which is separate from the main bacterial genome, only contains a few genes. So the way that we insert it then is the plasmid is cut open using the same restriction endonuclease that was used to cut out the gene of interest. That creates the same sticky ends, and therefore you're going to have sticky ends complementary to each other on the DNA fragment and on that cut open plasmid, and therefore we can join them together far more easily. Once then we have those complementary sticky ends, the DNA fragment has been cut to create that. We can then mix them together, and the enzyme ligase is used to stick together those sticky ends, sometimes known as annealing. And ligase is catalyzed in the condensation reaction to form phosphodiester bonds between the sticky ends of the plasmid and the sticky ends of the isolated DNA fragment. So now we've got our plasmid with the gene of interest inserted into it. We need to get that plasmid into the host cell, and that's what transformation is. So the vector, which is that plasmid with the recombinant DNA, next has to be inserted into the host cell, because that is the cell that's going to express that gene to create the protein. And in order to do this, we need to make the cell membrane of the host cell more permeable. And bacteria are your typical host cells. So to increase the permeability of that membrane to get the vector in, the host cells are mixed with calcium ions and heat shocked, which means a sudden increase in temperature. And that should enable the vector to enter the host cell cytoplasm. So that has now got the isolated DNA fragment into a vector and then into the host cell. But we do have to check that the host has definitely taken up a recombinant plasmid. So that's the next step in the process, identifying transformed cells, which means identifying cells that have taken up the recombinant plasmid. So not all of the host cells, which are usually bacteria, successfully take up the recombinant plasmid. And these are the three key issues that could occur. Maybe the recombinant plasmid just didn't get in through the cell membrane. Or maybe the plasmid that did get in is one that doesn't have the gene of interest in it. Because sometimes when you cut open the plasmid sticky ends just rejoin back together and you get your original plasmid again. And then sometimes the isolated DNA fragment of interest, that actually loops around and its sticky ends stick together to make a very, very tiny plasmid, and that gets inserted instead of the recombinant plasmid. So that's why we have to identify the transformed cells to make sure before we grow the bacteria en masse to create that protein of interest from the gene of interest, that you're definitely growing bacteria that contain the recombinant plasmid. So the way this is done is using marker genes. So marker genes on the plasmid is how we identify which bacteria has successfully taken up the recombinant plasmid. And the common marker genes that are used are marker genes for antibiotic resistance, or coding fluorescent proteins, or coding for enzymes. And that's because these make it easy to identify whether the bacteria has that gene in it. And therefore, if it does, it means it must have the plasmid within it as well. So another concept that you need to be aware of within this is that gene editing occurs, and this is a precise and targeted form of genetic engineering that involves the modification of DNA at specific locations within an organism's genome. And unlike traditional genetic engineering methods that typically involve the insertion of entire genes, gene editing allows for the precise insertion or deletion or replacement of DNA sequences at precise locations. And this precision is achieved using specialized tools such as CRISPR-Cas9, which acts as a molecular scissors to cut the DNA at specific sites determined by guide RNA molecules. So once the DNA is cut, cellular repair mechanisms can be harnessed to introduce desired changes to the genetic code. Gene editing techniques offer unprecedented control over genetic manipulation, and that allows scientists to modify genes with high accuracy and efficiency. This capability has numerous applications in research, in agriculture, medicine, and biotechnology, including the development of disease-resistant crops, the treatment of genetic disorders, and the creation of genetically modified organisms with tailored traits. The gene editing holds immense promises for advancing our understanding of genetics and revolutionizing various fields by enabling this precise and targeted modification to the genome. So you can also do in vitro cloning, which means not inside of the organism, and that's what PCR is. So it's a way to amplify the DNA fragments that you have. And once the DNA fragments have been isolated, they need to be cloned to create large quantities. We've already gone through how you do that in vivo in the bacteria, but in vitro is PCR, the polymerase chain reaction, which is all done in an automated machine. And in this PCR machine, you need a thermocycler, which is the machine, DNA fragment that you want to make lots of copies of, the enzyme DNA polymerase, and it's a special version of that, Taq polymerase. This Taq polymerase is taken from bacteria that are found in hot springs. So they're naturally adapted to survive at incredibly high temperatures. So the DNA polymerase, Taq polymerase, has a much, much higher optimum temperature, and it doesn't denature at your typical 37 degrees C. It denatures at much higher temperatures, and that enables the temperature in the thermocycler to be increased to speed up the reaction without denaturing the enzymes. Primers are needed as well to attach to the DNA fragments to enable the DNA replication to start, and DNA nucleotides are needed to create your new DNA sequences. So the method then, the temperature is first increased to 95 degrees C to break the hydrogen bonds between the two DNA strands. So we now have two single strands of DNA, and they both act as a template. The temperature is then decreased to 55 degrees C, so it's cool enough that the complementary DNA primers can align opposite those complementary bases and form hydrogen bonds to hold them in place opposite those complementary base pairs. The enzyme DNA polymerase will then attach, and the free-floating DNA nucleotides can align opposite the complementary base pairs. DNA polymerase will then catalyze the formation of phosphodiester bonds between adjacent nucleotides, and that's how we get the synthesis of the new DNA strand. That process is then just repeated. That's why it's called a thermocycler, because the temperature is changed continuously in this cycle. So 95, then 55, then 72 degrees C. And it's 72 degrees C for the final stage, because that's the optimum temperature for that Taq DNA polymerase. So PCR is fully automated, it's very efficient, it's rapid, you can make billions of copies of DNA within a few hours, and it doesn't require living cells like the in vivo did, which required bacterial cells, and therefore it's quicker and it's a less complex technique. We're then going to move on to DNA fingerprinting, which is a way to look at how closely related different individuals are based on VNTRs, which are variable number tandem repeats, which are found within the introns of DNA. And the probability of two individuals having the same VNTRs is very, very low, but the more closely related you are, the more similar your VNTRs are. And that is how genetic fingerprinting works. It's the analysis of VNTR DNA fragments to determine genetic relationships and genetic variability within a population. So here are the stages we're going to go through in genetic fingerprinting: collecting your sample of DNA to analyse, how you then extract just the DNA, digest it, separate it, and then ultimately analyse your sample. So collection, the smallest sample of DNA can be collected for genetic fingerprinting, and this could be from blood or body cells or hair follicles. And if the sample of DNA is really small, then PCR is going to be used first, so you have a large quantity. Then we need to digest this DNA, and we're going to use restriction endonucleases to cut the DNA into smaller fragments. And enzymes which cut close to target VNTRs are going to be added. To separate out the VNTR, those cut up fragments, the DNA samples are loaded into these tiny wells in a block of agar gel. The gel is then placed in a buffer liquid with an electrical voltage applied. DNA is negatively charged because of the phosphate groups, because there's a negative charge in that phosphate group. So the DNA samples will move through the gel towards the positive end of the gel. and they'll move at different speeds depending on how long the DNA fragment is, so how big that particular VNTR fragment is. And that is how it separates out. Now, the agar gel creates resistance for the moving DNA, and therefore smaller pieces of DNA can move faster and further along the gel. And this is how the different DNA VNTRs are separated. An alkaline is then added to separate the DNA from being double-stranded to single-stranded. And the hybridization stage is when DNA probes, which are short single-stranded pieces of DNA, which have been created to be complementary in terms of the base sequence of the VNTRs, those are added, and the DNA probes either have a radioactive or fluorescent label so that we can then move on to the development stage. But this part is called hybridization because the different DNA probes are mixed with those separated strands. So you've got two different pieces of DNA joining together, creating a hybrid. So the development then, the agar gel will actually shrink and crack as it dries. So we transfer the VNTRs and DNA probes using a nylon sheet. So transfer it onto that, and the nylon sheet can then be exposed to X-rays if you've used a radioactive probe, or you can expose it to UV light if it was a fluorescent probe that was used. And that is then how we get this development of your bands that we can actually see the positions of. So the position of the DNA bands are compared to identify genetic relationships, or it could be to see the presence of a disease-causing gene, or to match an unknown sample of DNA from a crime scene. And this is one that I actually created a long time ago. We can see this was the unknown that I had to identify. Here were the different samples of DNA that I had. And from this example, I could match that the unknown was the same DNA as number 3, because we can see the bands are in exactly the same position. And that is your analysis stage. So interpreting the DNA showing the results of the gel electrophoresis, this is your analysis stage again. And if you're going to do this in a paternity test, here we have the bands from the mother. Here are the bands from the child. And any band in the child that wasn't from the mother's DNA must be from the father's. And that is how you can use this in paternity tests. So you'd have to match up the bands to see which bands they have in common with the different fathers to see if that particular individual could be the father. You can also use microarrays, which are used to identify the genes present in an organism's genome and determine which genes are actively expressed in the cells. And this technology allows researchers to study a large number of genes very rapidly at the same time. So following the hybridization of DNA or an mRNA sample to the microarray, which is basically a plastic dish with lots of mini dimples in it, the data are then scanned and interpreted by a computer. And the process reveals which genes are shared between species or unique to one species or absent in both. It also enables the detection of gene expression patterns in specific cell types or conditions. So for instance, cancer cells exhibit different gene expression profiles compared to non-cancerous cells. And microarrays are used to compare gene activity between transcribed genes, converting mRNA to complementary DNA using reverse transcriptase, and amplifying that complementary DNA if necessary through PCR. You then get these fluorescently labelled complementary DNA, which hybridize with the probes on the microarray. And the fluorescent spots are going to indicate genes actively transcribed in the cell, and with the intensity of the fluorescence correlating with the gene activity level. This has been particularly useful in investigating mutations in genes associated with breast cancer, shedding light on the underlying genetic mechanisms of disease. So by providing the insight into gene expression patterns and activity levels, these microarrays offer invaluable information for understanding complex biological processes and facilitating advancements in genomics and biomedical research. Now, databases are also used, so research into the genes present in various organisms and their expression generates lots of data. And this data has to be stored digitally in a database. And that encompasses nucleotide sequences of genomes or individual genes, as well as amino acid sequences. And this field responsible for collecting, processing and analysing all of that information is known as bioinformatics. And software developers actually play a crucial role in creating these systems for storing, searching, retrieving and analysing this data. So computer technology facilitates the collection, analysis and also internet-based access to this information. Notable databases include Ensemble, which houses genomic data of eukaryotic organisms like humans and zebrafish and mice. UniProt, which contains information on protein sequences and functions. And the Protein Data Bank, which provides details on protein sequences and structures. And these databases are essential resources for researchers, providing valuable insights into genetics and protein biology. There are also selection and retrieval tools, such as the Basic Local Alignment Search Tool, or BLAST for short, enabling researchers to compare sequences and identify similarities with existing database entries. Comparison between sequenced genomes, such as the human genome with that of a fruit fly or nematode worm, has revealed evolutionary relationships and common ancestry. So it's been really, really useful. Model organisms like Drosophila are instrumental in studying gene functions and developmental processes due to how easy their genetics is to work with, but also how well characterised their genomes are. And that information from their genome helps us understand the human gene function and disease mechanisms as well. So although it's not humans we're working on, we still gain valuable information to help us understand diseases within humans. So this vast genomic information stored in databases has so many practical applications. So it's been used in malaria research, for example, where gene sequences have been utilised in vaccine development as well. Additionally, databases like CFTR2 provide accessible information on genetic variants associated with diseases like cystic fibrosis, and this helps healthcare professionals in diagnosis and treatment decisions. Then we go on to 19.2, genetic technology applied to medicine. So there are advantages of using this recombinant technology. Genetic engineering enables the production of recombinant human proteins, offering several advantages in the treatment of diseases. And here are just 3 examples of proteins that have been created using bacteria, yeast, or even mammalian cell cultures. And using these bacteria, yeast, and mammalian cell cultures, producing these human proteins has several advantages. First of all, these cells are uncomplicated nutritional requirements because they're very, very basic cells. You get large quantities of the desired protein being generated. That means you've got lots of this medicine available. You can do this in facilities that require minimal space because these are single cells rather than having entire multicellular organisms. The process can be conducted almost anywhere globally, and also the protein extraction from animals or blood collection from multiple donors would therefore minimise practical and ethical concerns because you're not having to do that, you're just taking it from a single-celled organism that is producing it. So other human proteins like the Factor VIII and the adenosine deaminase are produced using similar technologies. So we've got that Factor VIII is crucial for blood clotting, and it's synthesised by genetically modifying hamster cells. And by inserting the human Factor VIII gene into the hamster kidney and ovary cells cultured in fermenters, a constant supply of Factor VIII is produced, and that eliminates the need for blood donations and therefore the associated infection risk and the availability of the blood. Adenosine deaminase is essential for T lymphocyte function and can be produced using genetically modified bacteria, E. coli, or cell cultures derived from insect larvae. This enzyme therapy has been successfully utilised for over 2 decades to treat SCID, or severe combined immunodeficiency, highlighting its long-term efficacy as an alternative treatment option. Now, genetic screening is when you would have your DNA tested to screen whether you have the gene or an allele linked to an increased likelihood of developing a disease, or it could be to see if you will definitely develop a particular disease. So we've got a whole list of advantages here of why you might want to do this. You'd get an insight into an individual's genetic makeup. It might help you to prevent the development of a disease or get better management of it. It could result in the early detection of certain diseases, such as breast cancer, where early detection is essential for increasing survival rates. You can make informed decisions about your health. Early diagnosis in general can help with personalised treatments for certain diseases, such as Huntington's disease and cystic fibrosis. It will help with preventative measures. So certain diseases you won't definitely develop if you have the associated gene or allele, but if you modify your lifestyle, you're then going to reduce the likelihood of developing it. So overall, it can help to improve patient outcomes and also therefore reduce healthcare costs. Gene therapy is a way that we could try and treat genetic diseases as well. And it involves correcting faulty genes or introducing functional ones within the genome to override the affected cells. So for example, severe combined immunodeficiency and inherited eye diseases have been targeted for gene therapy interventions. So in SCID, where the deficiency is linked to ADA, which is comprising the immune function, gene therapy involves restoring normal ADA activity, and that will then give long-term benefits. In the inherited eye diseases like LCA, gene therapy restores vision by introducing functional alleles of the RPE65 gene into the affected retinal cells. And these examples highlight the potential of gene therapy in providing long-lasting solutions for genetic diseases, therefore improving patients' quality of life. Now, so far we've only highlighted all the positives with these technologies, but there are social and ethical considerations linked to this as well. So while genetic screening does empower individuals to make these informed decisions about their health, it does link to some concerns around privacy, discrimination, and also psychological impact. Because in terms of privacy, once you have screened yourself, there's the question of, well, who then gets access to that information? And it could be, for example, maybe insurance providers might get access, and therefore they wouldn't give you health insurance anymore because they know that you are at higher risk of developing a particular disease. You might get discriminated from an employer if they knew that you are likely to develop a disease later in life. And also the psychological impact of knowing you have an allele that makes you at risk of developing a disease. That can play heavily on someone's mind. So gene therapy does pose challenges related to safety, efficacy, and also equitable access to treatment as well. The ethical dilemmas arise regarding the manipulation of human genetic material and therefore the potential for eugenics and the allocation of resources. So we do have to strike a balance between the benefits of these scientific progresses whilst also considering the ethics and the social side of the considerations as well. And lastly then, it's genetically modified organisms in agriculture. So genetic engineering is one technology that could really help with the global demand for food because we could enhance the quality and productivity of farmed animals and crop plants. So for example, GM salmon have been developed to grow faster and more efficiently, offering a potential solution to meet the increasing demand for seafood. Also, we've got herbicide-resistant soybean varieties have been engineered to tolerate herbicides, which enables farmers to control weeds more effectively and therefore increase their crop yield. We've also got insect-resistant cotton varieties created by incorporating genes into Bt bacteria, which produce toxins lethal to certain pests, and therefore it would kill the pests without needing to apply chemical pesticides, and therefore it's a more sustainable agriculture. So this shows how you could use genetic engineering to improve food security, sustainability, and also economics. But again, there are downsides to consider linked to the ethical and social implications. So the use of genetically modified organisms to produce food in this way has one issue around GM cultivation. So unintended effects maybe of non-targeted organisms. So maybe it does actually affect other organisms, not just the crop plant of interest. It could result in maybe biodiversity loss if it causes the death of other plants. And also you might get herbicide resistant weeds and pest populations. There's also concerns that this is a relatively new technology, so we don't know the long-term effects of humans eating GM food. And there may well be no long-term effects, but we actually don't know because we haven't tested it. We've got social economic factors as well, because these seeds are very expensive. So therefore, they become too expensive for poorer farmers or poorer countries to be able to afford to buy these seeds to get these GM crops. And then we've got ethical debates as well, which is looking at the moral implications of manipulating living organisms and the potential consequences it might have on animal welfare, ecosystem integrity, and future generations as well. So that takes us to the end of the genetic technologies topic. I hope you found it helpful, and if you did, don't forget to subscribe.